Dr. Cindy Bachurski is the Director of Pipeline Programs in the Office of Academic Affairs and Career Development at Cincinnati Children's Hospital, and her presentation today will cover biomedical research career paths. Dr. Bachurski?
Thank you, Jamila. I'm happy to talk to you students today, and I'm going to be talking about not only biomedical career paths, but some opportunities that we have at Cincinnati Children's to get you involved in research, both in high school and as an undergrad, as you move forward in your thoughts and your career paths. So I put this graphic up here because I think it really tells a lot about the kind of qualities that you need to be a scientist. You need to be somebody that observes and wonders, somebody who's good at sharing your ideas and discoveries. That's one of the fun parts of being a scientist. You listen to others, you explore the world, you ask questions. You have to be somebody that has lots of questions. You conduct experiments to try to answer your questions, and you use all kinds of different tools to solve the problems. So it could be you in the future. So these are some pictures of what a scientist looks like in the media. So this is, I think, is Dr. Brown from Back to the Future, and somebody who's all dressed up in some crazy outfit here, which is not really what scientists look like. Here's some pictures of some scientists at work at Cincinnati Children's and the College of Medicine. You can see the one big thing that they have in common is they're usually wearing a lab coat, usually wearing a long lab coat. So what is the research at Cincinnati Children's? So the Children's is one of the largest research institutions in the country for pediatric diseases. It has over 1.4 million square feet. And if you have gone to Cincinnati Children's as a patient, you probably don't even realize this, but there's a picture here in the bottom where the hospital is in the front and the research building kind of towers behind it. And it goes back this way as well. So this is a couple different pictures of our research buildings. They're huge. We have lots of researchers. We have over 50 academic departments, 23 research cores. And one of the big things about Children's is the interdisciplinary research.
There's a word people throw around a lot at Cincinnati Children's: collaboration. When you get here, you'll understand it's not just a word, it's part of our culture. Science is a group effort here, and we work together with the University of Cincinnati, both in the lab and at the bedside, to train the next generation of investigators and physicians.
Coming to Cincinnati Children's, I was really struck by how interactive and collaborative an academic environment it is. In fact, I would say it is exceptional in that regard.
Collaboration is a huge part of what my group does. We study the DNA, the genetic etiology of different diseases, and it's exciting when we get to collaborate with another group that looks at the same disease from a little bit different paradigm.
The immunology graduate program here.
Okay, this is a very long video. If you ever want to see it, you can go to Cincinnati Children's Immunology graduate website and see the full thing. But just to give you an idea of what the collaboration is like at Children's, that there's very close interactions between both doctors and scientists. So these are some of the research areas that Children's focuses on. You can see from everything from developmental biology, how the baby forms, to neuroscience. I think earlier this week we had some neuroscience researchers talking. We have genetics, stem cell biology, lung biology, imaging research, even epidemiology and health disparities. So there's a lot of different areas of research that you can go in at Cincinnati Children's. So some of the major model systems that are used at Cincinnati Children's for basic science research are listed here, including microorganisms. We even do research on the new virus that causes COVID-19. So here are some terms that I will be using a lot today and maybe have already used that you may not understand the difference between. So there's basic science research, translational, and clinical research. So basic is where you're just trying to understand the basic biological processes and the genetics of normal and disease processes. Translational research is where you're trying to put that link between the basic research and treating human diseases. And then clinical research is where you're looking at either drugs, diagnostic products, or some kinds of treatment like the current trials for the COVID-19 virus. So just to try to figure out how to treat the diseases that you've learned through your basic and translational research. So there's some pictures here. On the left is a basic science researcher looking at cell culture in a dish with a microscope. And on the right, there's a clinical researcher talking to patients. So some examples of this kind of research that we've had this week would be Jennifer Pritchard-Cram talked about basic science. Matthew Beatty is the biomedical engineer that talked yesterday, and he provides help with both basic and clinical research. And then we've had a lot of people talking about some of the different areas of clinical research. Paige is a graduate student doing clinical research. Andrea Meisman talked about the clinical research coordinator career path. Suzanne Summer talked about nutritionists and the metabolic kitchen and how that supports clinical research. And our final speaker today, Mary Staat, is going to talk about her research in viral diseases. So as you progress through your career and you're as a student, we have lots of opportunities here at Cincinnati Children's. Undergraduate students and high school seniors can participate in the BRIMS program. We have a summer research program. We have co-op research opportunities here. Once you get your bachelor's degree, there's a lot of choices you can make, and you can participate in research directly with a bachelor's degree. And there's two research paths on the right here. You can either be a research assistant working in basic or translational research, or you can be a clinical research coordinator, which we, as I said, Andrea talked about this whole career path. And there's a career path. You start with a bachelor's degree, you're a research assistant too, and then you can progress to a 3, 4, or a senior research assistant. Or if you start as a research assistant, you say, hey, you know, I think I have a lot of questions that I like ask my own questions instead of somebody else's. You can go to graduate school. We have lots of graduate programs that students working here at Cincinnati Children's. Our PhD and MD and MD PhD programs are all options for you as a student at the University of Cincinnati College of Medicine to then do your research at Cincinnati Children's. So I'm going to talk now about, so what do some labs look like? What does it look like if you're working in a lab? What kind of people do you work with? You're not working alone. As we said, it's very collaborative. So this is just an example of Dr. Sumanis' lab. He's a basic science researcher. He studies blood vessels and also aneurysms, which is when your blood vessels burst. And he uses a model of zebrafish. So this is a cool thing about zebrafish. When they're an embryo, and this is an embryo that is almost at the fish stage, this pink part here is the yolk sac. But you can see the green, all of the blood vessels in this fish are fluorescent. So you can actually, as the fish matures, it goes from egg to fish in one day. So you can actually do stop motion photography and watch all the cells form into the blood vessels. It's a very cool model system, and he's got a very powerful system going on there. But you can see he has lots of different kinds of people working for him. He's got a research assistant, like I just told you, who has a bachelor's degree. He's got several undergraduate students working with him. He has several graduate students that are working on their PhDs. He has somebody who's called a postdoc, which already has his PhD and is working on additional training. And then he has a research scientist that kind of oversees everybody. She's a more senior PhD level scientist that works with him. So here's another kind of lab. This is a neuroscience lab, Dr. Matt Skelton. He studies brain disorders and metabolic diseases that cause problems in your brain. And he's a much smaller group. Marla is a postdoc. Zuhair and Kayla are both graduate students, and Amanda is a research assistant. And they do behavior studies with mice, and with mice that have particular diseases, this creatine deficiency disease. So I'm going to focus a little bit on Kayla. She's one of our graduate students here. She's from Alabama. She got her bachelor's degree from Oakwood University, and she is working in the neuroscience PhD program. She's almost finished now. She's been working on this creatine transporter deficiency, which is a pretty rare disease that causes intellectual disability in men only. So it's an excellent trait. So she's investigating the use of what they call as a ketogenic diet. So how to use diet to treat a disability. So I wanted to show a little bit about what the timeline is to get a PhD. So usually what you need to do is undergraduate research, and I'll talk a little bit more about how you can get that. Once you get your bachelor's degree, you come and join a PhD program, and you work for 4 to 6 years. The first couple years are on additional class training, and then the rest of the time is on your research. At Cincinnati Children's and the College of Medicine, you get paid the whole time, so it doesn't cost you anything, and it's a living wage. After you finish your PhD, then you would go on and do a postdoctoral fellowship in something else. So she works with mice. So here's another example of a more complicated lab. This lab is a clinical and translational research lab. They're looking at the genetic basis of growth disorders. So we have Dr. Bachurski is an MD doing research, and then Dr. Hua is a PhD, and they work together. So this is what they're studying. He is an endocrinologist. So an endocrinologist is somebody that studies diseases of hormones in your body. Some of the diseases that endocrinologists might treat are diabetes, thyroid disease, obesity, or growth hormone defects. And what happens when you don't have enough growth hormone? You're short. Growth hormone makes you grow. So it's diseases of short stature. So he has several families. Many of the genetic mutations that cause short stature are known, but not all of them. So if he's got a family that doesn't have one of the known mutations, what they will do is they will sequence the DNA from the whole family to try to figure out what the difference is in the patient with short stature, say, versus a brother or sister who doesn't have that problem. Once they find some potential defects, what they will do is then Dr. Hua's lab models that in mice by changing the DNA in that same gene that a mouse has and see if it makes a small mouse. Once she has this figured out, then she can use that mouse model to figure out how to treat patients and potentially overcome the short stature problem in the future. So one of the students that works in that lab is Corinne Foley. She's getting both an MD and a PhD at the same time, and you can do that. It's called an MD PhD program or an MSTP program. So she's already completed the first 2 years of medical school, and she's now working on her PhD. So she's interested in nutrition, pediatrics, education, and rare genetic diseases, and that's why she joined this lab. So in order to get into the MD PhD program, first you need a bachelor's degree, you need to have significant undergraduate research, and you need to do very well in the MCAT exam, which is the exam that allows you to get into medical school. After the first 2 years of medical school, you pass your first board exam, then you're in that PhD mode, you get your PhD, you finish that, then you go back and complete the last 2 years of medical school. That's the clinical years. You pass your second board exam, then you're an MD and a PhD, but you're not done yet. You still have to do residency or an internship, and most MD PhDs specialize, so they have to do a fellowship program as well, and then they usually get a faculty position. MD PhD is not for the faint of heart. It takes a long time, and you have to really know that you want to both treat patients and do research to understand how diseases work. But it's really great because if you're in an MD PhD program like the one at Cincinnati Children's and the College of Medicine, it will pay you the whole time, and you come out after all that time having been paid and not have any debt. So that's a nice thing, but that's not a reason to do it. So now the last two people I'm going to talk about are both clinical researchers. So Dr. Modi studies adherence to treatments. So she's a clinical psychologist, and she tries to study what is it about treatments that some kids that have diseases that are just chronic and they need to be taking medications their whole life, why don't they take their medicines? And she particularly is studying epilepsy. She has a clinical lab, so she has clinical research coordinators, as I said, like Meisman talked about earlier. She also has some graduate students in getting a PhD in psychology. She has a postdoctoral fellow, and she takes undergraduate students in the summer as well to help her with her studies. Here's, we had another person earlier this week talk about sports medicine as an athletic trainer. Dr. Kiefer is a PhD in sports medicine research, and he actually uses virtual reality to help with identifying children that have had concussion after a sports injury. So we had a student in our BRIMS program work with him one summer. She actually got a prize for her poster, and she was working on trying to compare the virtual reality approach with the conventional eye tracking test for concussion. Actually, they're using virtual reality now. That was a couple years ago. So I'm now, the last part of my talk, I'm going to talk about the opportunities that we have at Cincinnati Children's for research and better understanding of research career paths. So first, you're here in the Middle School Science Symposium, 7th and 8th grade. So that's for middle school students. We also have a high school science symposium. We have job shadow programs. We have a senior summer internship program and a BRIMS program for high school seniors. And then once you're in college, you can also join the BRIMS program as a freshman or the SURF programs. I'm going to talk about all these programs pretty quickly in the next couple slides. So first, the job shadow program. This is something that anybody can do as long as you're a minimum of 16 years old. We offer this program once a month, all throughout the year. We take about 20 people each month, so the program fills up very quickly. You get to do about a 3 to 4 hour job shadowing of a professional in many different career paths, including doctors. You have to sign up online. Unfortunately, during COVID, it's suspended. So I would say, look next summer. Well, you guys are probably too young anyway. So by the time you're 16, you'll be able to do it. The High School Science Symposium is an awesome opportunity. You should definitely try to participate in that by the time you're in high school and a junior. COVID will be a thing of the past. So you'll be able to come and both hear a little bit more in-depth allied health and career path talks. We also have great group and interactive presentations. You'll get tours all around the research side and the clinical side of Children's Hospital. And then you can have lunch with specialists in all different areas, including researchers. The summer internship program is for graduating seniors who are considering either an MD, a PhD, or that MD, PhD career track that I was talking about. It's an 8-week paid program. You're working at Children's Hospital for 20 hours a week in the summer. You get matched with a clinical faculty mentor. Some of those mentors actually do research, so you could potentially be in a research lab, or you would be just mostly doing clinical shadowing. We also have rotations in other specialties and luncheons with specialists all week. And you actually get to do some hands-on stuff. This is not a real person. This is a mannequin. So that's one of our ways of training people. But we do have 3 surgeons that allow students to shadow that are mentors. There's another one of our surgeons that's a mentor. At that undergraduate research or pre-med students. Pre-med students get to do research, either clinical research, translational research. Some pre-med students even do basic science research. It really helps you to understand how that whole medical process works. So first is the BRIMS program. This is for Cincinnati area students only. Applications are due in March. It's for graduating seniors or college freshmen, and you have to be in an underrepresented group, either African American, Hispanic, Pacific Islander, or American Indian, or Native Alaskan Natives. We don't have too many Alaskan Natives here, but we have all the other ones. So definitely you want to Get your applications in. This is an excellent, excellent program. It's 9 weeks full-time paid research. You get to do your own research project. We match you specifically with the kind of research that you're interested in. We have all those opportunities that I was just talking about available. And then you are part of the SURF program, and you participate in the capstone poster competition. So really the requirements are you need to have a GPA above 3.5 and really have an interest in research. Here's some pictures of BRIM students. These students are actually working on an asthma project. And so this is actually human samples that they're freezing down here. This student was working with a mouse model. This student was actually working on biomedical informatics. He is just working on computer programming. There's a lot of different opportunities, as I said. So here's a quote from one of the students from the 2011 BRIMS program who then went on, got his MD at the College of Medicine, and is now a resident doctor. I don't know why that's going forward. It was a truly transformative experience, instigating a curiosity for research and passion for medicine that led me to where I am today. So BRIMS program is awesome. Another really awesome program that we have in the summer is the SURF program. The SURF program, while the other programs that I talked about are all for students just from the Cincinnati area and there's no housing available, the SURF program is actually a nationally competitive program. It's much bigger. There's almost 200 students every summer in the SURF program. This is 10 weeks of full-time summer research. Again, you're doing your own research project in one of our world-class laboratories. There's a lot of professional enrichment opportunities that we offer as well to the students. We pay them a stipend. Right now it's $400 a week. It's probably going to be more by the time you're there. And there is housing available. So you would be with students from all over the country, including Puerto Rico. We've had students from Hawaii even. So this is just a little overview of what the events look like in SURF, other than you're working 40 hours a week. We have social events. We have some biomedical kind of presentations, including an introduction to surgery. And the surgeon that gives that presentation allows students to shadow him as well. We have all kinds of talks. And then we have professional development. So we have presentations on scientific writing, research ethics, how to design experiments, how to apply to medical school and graduate school, help with your resumes and networking. And then again, the big capstone poster symposium. So here's some pictures of students working on their projects. And then this is a picture of what the capstone poster symposium looks like. Each student makes a poster, they stand in front of it, they get judged, they have to present their data that they generated in the summer. This is picture down in here is Emily. She did work with neuroimaging of kids hearing things, and that's why she's pointing to her ears, because she's talking about hearing. So I would like to end with just a couple things for you to think about if you're thinking about maybe becoming a scientist. First of all, it's really important to cherish your family and friends, and then to think about, know yourself. What makes you happy? What do you love doing? What inspires or motivates you? What are your unique gifts? What are your challenges? What do you really need to work on? And in all of life, either love what you do or change it. It's your life and your decision. Learn from everybody around you. You want to always do your best, and really the future is your choice. Thank you for listening to me, and I hope you enjoy the rest of the day.
Thank you so much, Dr. Bachurski. Dr. Mary Allen Staat is a professor and member of the Division of Infectious Diseases and also the director of the International Adoption Center at Cincinnati Children's. Dr. Staat's presentation will give an update on the COVID-19 pandemic and answer any questions that you may have. Dr. Staat?
Well, thanks for inviting me to be here. I love talking about what I do. I really feel like I have the best job in the world at Cincinnati Children's, and so excited to tell you about kind of my career path, and then also to end with some COVID-19 mythbusters. So just to start out, to tell you a little bit about my job at Cincinnati Children's. First of all, I've been here for 25 years. Last month was my 25th anniversary here at Children's. And I came here as what we would call junior faculty. It was my first real faculty position. I really just have lived out my whole career here. I'm a physician, so I'm a medical doctor. I have an MD, a medical degree. I'm a researcher, and as such, I went ahead and got my master's in public health, which I'll tell you a little bit about, to kind of help me to do better research. And then I also am an educator, and I'm an administrator. So I administer, lots of different programs here at Children's. So I was asked to tell you a little bit about the day in the life for me, and every day is definitely not the same, and that's really what I love about it. I have clinical duties, I have research duties, I get to educate, and as I mentioned, run different programs. Okay, so clinically, I get to see children with serious infectious diseases, and it's what we call being on call. Okay, and so I'm on call in the hospital to take care of kids that have really unusual infections. They may have infections like a brain infection or a heart infection or a bone infection, not your typical run-of-the-mill kind of cold or anything like that, but really serious infections. And I do that 6 to 8 weeks out of the year, where I'm that main doctor that goes and sees all the consults in the hospital who have these types of infections. I also take care of children who are internationally adopted, and I do that 1 full day a week in the outpatient setting. So I see kids in clinic who are internationally adopted. And then I also take care of people who are traveling abroad to give them travel advice in our travel clinic. And I do that about 1 day a month. My research focuses on epidemiology and vaccine safety. So I'm an infectious disease doctor. And so all of my research is really around various different infectious diseases in both children and adults. And for education, I do lectures for medical students. We also teach while we're taking care of patients on what we call rounds. And we do other lectures for clinical and research topics, both like here at Children's or at UC, but really across the United States and even internationally at times. And then, as I mentioned, I'm an administrator, which means I am kind of the boss of these different clinics and programs. So I created the International Adoption Center a little over 20 years ago, as well as the International Travel Clinic about 10 years ago, and I run those. And then I'm also the director of our epidemiology, surveillance, and vaccine safety program. So what did I have to do in order to get to this position? It's really, I actually think I've put out of my mind how many years it took to do this, but anyways, many years, okay? So don't let it be daunting, because the school, going to school was really so fun for me. I really actually miss going to school and learning in that formal way. I went to college, and that was for 4 years, and got a bachelor's degree, then med school for 4 years and did an MD degree. I did a residency in pediatrics. So once you finish medical school, you decide what specialty you want to go into. So I did pediatrics, and that's a 3-year program. And then if you want to subspecialize, you have even more special area outside of pediatrics, such as infectious diseases or cardiology or gastroenterology, you do a fellowship. And so I did a fellowship in infectious diseases for 3 years. And then, as I mentioned, I went ahead and got my master's in public health, which took 1 year. My path, not exactly like that, but I'll take you through it. I actually grew up here in Cincinnati. I left when I was 17, and I thought I'd never come back, and then I really couldn't wait to come back after I'd done my training. I went to high school at Lloyd Memorial High School in Erlanger, Kentucky. I then went to college at George Williams College outside of Chicago, and I majored in physiology, and I really gained an interest in international health while I was there, and I went to Suriname and studied food behavior in pregnant women. So really, right at the beginning, I was very inquisitive and had this research idea in my head, and did it even when I did a summer program abroad. Then I went to med school at University of Kentucky, and I found that I was interested in pediatrics and public health. I had this one terrific instructor, a physician who was in preventive medicine, who really inspired me to think about medicine differently and to think of it more globally with a public health focus. And he really influenced me tremendously, because my whole path took off in a different direction after having that one course with him. I continued with my interest in international health, and I traveled to Ecuador and did some clinical work there. I then did my pediatric residency and infectious disease fellowship at Baylor College of Medicine at the Texas Children's Hospital. And it was really a wonderful time. I loved going someplace different than the region, where I'd been, grew up and studied. And I love the international flavor of Baylor. It really has a very diverse population. I got to improve upon my Spanish skills. Got to see lots of different types of infectious diseases and conditions. And it was really a great learning experience. But you can choose to do your residency in your infectious disease fellowship really anywhere. You apply to those programs, and then you interview and then get accepted to one of those programs. And you don't have to do your residency and your fellowship at the same place. You can go to different places. Then as I mentioned, I really wanted to get some extra training in public health and epidemiology. And I wanted to do that because I really, again, had that interest in both international health and public health, but I thought I wanted to do some research. And so it's really helpful to have kind of a little bit more training than what I had in college, in med school, in order to do good research. So I went to Hopkins to get my master's of public health. And then I decided to go ahead and do a preventive medicine residency while I was there because I love to go to school and do training programs. So I did epidemiology and international health focus at the School of Public Health. And I think more people know about epidemiology now than they ever did. But most people, if you ask them on the street, they really wouldn't know what it is. But it's really the study of disease in populations. And COVID is a great example of how we're really using epidemiology to better understand this new pathogen. But it's really a framework for what we a framework for learning about all types of infectious diseases and other conditions, not just infectious diseases. So I got my master's in public health and did this extra training in preventive medicine and public health. And just a reminder, our public health thank you day is November 23rd, 2020. So remember to, first of all, know that there are public health people out there that are working to make everybody safer and have better health in the United States and abroad, and to thank them. I did my epidemiology fellowship at the National Institute of Allergy and Infectious Diseases, and you might recognize this fellow with the face mask. He was actually my boss, Dr. Fauci. And so it's been very exciting for me to see, I mean, I've always known how terrific he was and how big an impact he's made, in the field of infectious diseases, but really to see him, on the forefront with the COVID epidemic to really, see what a difference he's making there too, with really good, sound scientific information that he's presenting. So as I mentioned, my first faculty position was here, and I came to Cincinnati in 1995. And one of the first things I wanted to do was to develop the International Adoption Center. And I did that because we had just adopted our kids, and I recognized that there was really a need for people to get some extra help who were going through the process. So those are my three kids there, George, Emily, and Katie, when they were little, and then more recently. And I did this out of it certainly wasn't in my job description, but it was something that I was permitted to go ahead and explore and develop here at Children's. And I really just wanted to help other families. And so again, we're just at 21 years this year of having this International Adoption Center, and it's really been such a passion for me and so wonderful to be able to take care of these kids and families. But really the reason Children's brought me here and recruited me is because they wanted me to develop a research career in surveillance systems for infectious diseases and help them with epidemiology. So I thought I'd spend a little moment here to talk about our infectious disease and vaccine research. And you've probably heard from lots of other different folks about all these different areas that you can do research in. Basic science, clinical trials, vaccine development, and epidemiology. And maybe you haven't heard much about epidemiology, but it's really a key component of understanding infectious diseases and developing vaccines. And we just have this is just, there's other pieces of the puzzle, but those are the main pieces of the puzzle for us when we're looking at infectious diseases and vaccines. So how does the epidemiology fit in? So before there's a vaccine, you need to know, is there just a need for the vaccine? Well, it's pretty obvious right now with COVID that there's a need for something to help prevent COVID from spreading and hurting people and causing people to be ill. But for some diseases, there's often a question. It's like, do we really need a Lyme disease vaccine? Do we really need a vaccine for, para-influenza? Do we really need that vaccine? And to answer that question, we really want to understand the burden of disease. We want to understand how much disease is out there and who is affected by this disease. What ages are affected? Do we need a vaccine for just young kids and babies, or do we need it for adults as well? So we have to look at the whole spectrum of how that infection impacts a population. Are there groups that are high risk? Are kids like with cancer at higher risk, or people that have diabetes at higher risk? Understand those special populations so that we can even target them to get a vaccine that'll benefit them even more. And then what are the costs of the disease? Nowadays, we're always looking at cost benefits. Is it too expensive to have a vaccine for the little bit of disease that it'll prevent or not? Okay. And then after there is a vaccine, is the vaccine safe? Is it effective? And what's the impact? So we do all these very nice vaccine trials in very select populations and healthy, typically white individuals of a certain age without any underlying diseases. And then we gradually expanded out to other groups, to kids, to the elderly, to people with more high risk conditions. And we give those vaccines in the community where it's not as controlled. And so once it's out in the community and it's being used routinely, is it still as effective, or does it lose some of its effectiveness because it's not being given in that very standardized approach? So my lab is actually just Cincinnati Children's, okay? I utilize all the resources that are at Cincinnati Children's to help me do my research. And my population is the greater Cincinnati area, and specifically Hamilton County. So what's so special about Cincinnati Children's in Hamilton County? This was the one thing that really, I think, helped my career out, is I recognized that Hamilton County was just large enough to study almost any condition that was serious enough to bring you to the emergency room or to be admitted. The number of births is about 10,000 a year. It's really a very stable population with little in or out migration. So like once you live in Cincinnati, there's not very many people leaving or coming into Cincinnati. That's very different than a lot of other populations. And really importantly, Cincinnati Children captures nearly all Hamilton County children who were hospitalized or seen in the ED. So we know exactly how much severe disease we have. Our other partners that we do surveillance with across the United States, they usually have to do 2 or 3 other hospitals in their city in order to identify all the kids that have flu, or identify all the kids that have chickenpox or COVID for that matter. So, we capture almost all young kids, certainly kids less than 5 here at Cincinnati Children's, and that makes us very unique. It allows us to calculate population-based incident rates. Okay, so that means that we're looking at people over a year and how many of those people in our population get that disease, get flu, get COVID, get whatever we're studying, okay? So it's a rate. It's like a certain number per that population for that year, as opposed to just taking a snapshot at any one time. If you looked at COVID over the summer, it was less, it was probably 1% of people. If you did a prevalence, meaning just at that one snapshot in time, how many people had COVID, that tells you one story, okay? But now, you may have 5, 10, 15% of a population that is positive. Okay, so, but what does it look like for the entire year period? And that's really what we want to know most of the time. So I have a couple slides here to show what we can do with these data. Rotavirus, many of you all probably got the vaccine, or you might be just a little bit too old to have gotten the vaccine, but you got the vaccine to prevent rotavirus, which is a diarrheal disease that young babies get and can get very sick from. They usually will not die from this in the United States, but across the world, many children actually died from rotavirus, okay? So we wanted to develop a vaccine, and just a fun fact is one of my colleagues in my division actually developed one of the vaccines that eventually got licensed, Rotarix. And there's another vaccine called Rototec that got licensed for use as well. So first of all, we wanted to understand, how much disease burden is there? And as you can see on that bottom graph of the rotavirus seasonality, that in fact, there was a fair amount of cases, okay, that we saw in 2006 and 2007. Right that year, in 2007, we started having rotavirus vaccine introduced, and you can see the dramatic decline in 2008 that happened after a vaccine was introduced to young babies. We had almost no disease that next year, okay? The following year we had a little bit, and then we didn't have the next year. And then you can just see this every other year seasonality and what we call a biennial trend, where every other year you see a little bump in cases, okay? And that's kind of, a nice, simple way of thinking about epidemiology. It's looking at how much disease there is, how it changes over time, and is there, specific months out of the year where you see it. You can see on that graph, on the rotavirus seasonality graph, that, we can look at it by hospitalized kids, emergency department kids. It just gives us a picture of what's happening with a certain disease. On this slide, here to the right, we look at rotavirus trends over time. Again, it's kind of the same time period, but now we're adding more layers to it. We have the same kind of graphs. Now we're doing rates of disease. So we're standardizing it to the Hamilton County population. Here we have rotavirus ED visits. Here we have rotavirus hospitalizations. And you can see that there was high rates here and then a dramatic decline. And then every other year, we have a little peak, okay? But nowhere near what it used to be, okay? And then you can see here, this line, this blue line, shows you the vaccine uptake. And you can see that as vaccine uptake increased, we had this big decline in cases, in the incidence, and we've maintained that, okay? So that's been a terrific success program, and these vaccines have saved lives around the world. So it's been so exciting for us to be a part of this with the epidemiology, because a lot of what we developed with our surveillance program and trying to look at testing all these kids that come in the hospital for rotavirus to see what happens over time, those protocols got expanded to really around the world and are used to this day to monitor rotavirus disease. We use these same methods of surveillance, of looking at all the kids that come in the hospital, all the kids that come in ED with certain symptoms, to do surveillance for flu, to do surveillance for acute flaccid paralysis that's associated with an enterovirus, for COVID. So it's establishing kind of a system that you can then expand to other diseases to understand what's happening in your population. We also look at cost of the disease. We look at risk factors for disease. So are the rates different for Blacks or whites, Hispanic population, for older kids, younger kids? And then again, not showing the data here, but we also look at how effective the vaccines are. So once the vaccines are in the community, are the vaccines just as effective as they were in the trials? And most of the time, they're not going to be quite as effective, but they are still highly effective in almost every case. But they're going to be a little less effective because you have all those other variables that are not controlled for. Okay, this is a really fun study that we got to do, and I'm just using it to illustrate rotavirus vaccine safety. And so I mentioned about that rotavirus vaccine, which is a great vaccine. It doesn't have very many side effects, but in a small percent of kids, for whatever reason, and we don't quite understand why, they get something called intussusception, okay? And this just shows you that here's your intestines, and what happens is this, here's your large intestine, here's your small intestine, and for whatever reason, this small intestine kind of telescopes up into that large intestine and causes a blockage. And here you can see it on an X-ray that it just shows this little indentation here, where during the normal course of the peristalsis, that little small intestine slips up into the large intestine and gets stuck and can be quite serious. Kids can die from it, okay? And this is pretty cool. This shows you actually in the operating room how they are taking apart this intussusception here, just manually pulling it apart. We were asked, and we competed for a project to actually enroll 6-week-old babies, okay, to study them giving one of the 2 rotavirus vaccines that I talked about to see if their intestines behave differently with one vaccine compared to the other. So you can imagine we approach these little 6-week-old babies, moms, to see if they want to have their baby in this study. We had to do an MRI of their intestines to see what their intestines look like with this vaccine being administered to them, a few days after the vaccine was administered to them. We gave them something called barium that makes their intestines kind of light up to be seen on the MRI. We had to strap them in a papoose, and then we had to protect their hearing because they were going into an MRI. Okay, so we had 3 layers there so that the MRI sounds wouldn't hurt them. Here our little guy is cozy and ready for the scanner. In fact, they loved the barium. They fell asleep right away. And we stuck them right in that machine, and they were in there for almost an hour and just slept right through it. And it was really one of the easiest studies we ever did. And here's what we got after the finished MRIs. We got these beautiful pictures that showed how the intestines looked, and then also how the intestines moved. And we in fact found that there were really no differences between the 2 vaccines with regard to how the intestine looked and moved after finishing the study. I also want to mention something about our clinical research, okay? So this is a little different. Sometimes we will be just clinically curious about things that we're doing or seeing in our work. And that was certainly the case with international adopted kids. We are, seeing these kids over the years. We've seen almost 4,000 kids over the 20+ years that I've been here. And there are some patterns that you were seeing, and it's great to see patterns, but you really want to do some research to help you see if your instincts are right, that there is something really going on, and then importantly, if there is, and that would result in a change in guidelines to how we can better care for these kids, we want to go ahead and study it and publish those results. And this is just an example here. We thought hepatitis A vaccine is a vaccine you probably have gotten that is routinely given, but years ago it wasn't routinely given, and we wanted to understand whether or not these kids that were coming home from their birth countries needed the vaccine. Hepatitis A is like something that it's a virus that's transmitted through food and just like a contaminated environment. And we knew that our kids were really, who were in orphanages and these resource poor countries were at risk for having hepatitis A, and they may not even need that vaccine. And in fact, you can see the older the kids are over time, when they came home. So this older group of kids, when they came home, 90% of them already had protection. They already had hepatitis A antibody and would not need that vaccine. So that's a vaccine that we could not have to give them, okay? And most of our kids that come home have to get lots and lots of vaccines to get them caught up on a US schedule. And so if we can avoid giving them vaccines that they already have had or have had an infection from and they don't need, then that's really great for those kids and for the families. If you know me, which you don't, but I tell you, I love parasites, okay? And here's an example of a study that we did to better understand kind of how many kids got Giardia, which is a cute little parasite here. And this is Ascaris, the biggest worm that a kid might pass. There's lots of worms that they can have, but this is kind of a big one that they could have. And what we want to know is how many stools do you really have to look at to figure out if they have any of the infections. And so what we did was we had the families turn in 3 different stool specimens, and then we measured how much more increased yield did we have with each subsequent stool specimen. And so each of these, this is any pathogen, and this is like Giardia, this little organism here. It's another parasite, another one. This is the Ascaris here. And this is a dwarf tapeworm here. And if we just turned in 1 specimen, you were 62% likely to find it. But if you turned in even a second one, you were 95% likely to have it. And then all of them, of course, we stopped at 3. So this was really, really helpful to how we now manage kids that are internationally adopted. And it impacts care beyond just what we're doing. So we want to, take care of our families and our kids to the best of our ability, of course, but we really also want to translate what we are learning from our research to help other kids, around the world. And so these are just some of the things that I write for and provide, our data for, to develop guidelines and chapters for various things. And importantly, like for all the international adoption, we have chapters in all of these different books that lay out kind of how to best care for internationally adopted kids. And it's because we did this clinical research. Okay, so you heard a little bit about our infectious disease epidemiology and surveillance program. So as you can imagine, we really had systems in place to study kind of any infectious disease that might come into our community. So we were really well positioned to study COVID-19. And, we immediately, once we heard that it had, come to the United States, we started setting up studies, writing projects, protocols to study COVID in moms, in children, in our employees, in our daycare workers, foster care kids, pretty much any population that we could think of that we could then impact by providing knowledge to everyone about these populations. We also are doing studies on household transmission. So if someone's positive, we bring the whole household in to see how many people in the household have it. We check their blood. We then also look at their immune response to their infection. So we not only swab their nose to see if they have it, we then also, get saliva, we get stool, we get cheek swabs to look at their genetics to see if they're, something particularly interesting or important about their genetics that keeps them from getting sick or makes them get sicker. And then we do these immediately when they're sick, but then we do what we call convalescent specimens, so later down the road, to see how long that antibody actually lasts. That'll be really important with COVID because we really don't understand antibody responses very well yet. And then other folks in my group are doing vaccine trials, and then I'll be working as soon as the vaccines are out to be monitoring vaccine safety and to be looking at vaccine safety in special populations like kids and moms. So I want to end with our COVID-19 Mythbusters. And just a little disclaimer, I'm doing this talk in November, so who knows what we're going to learn. And I hope we learn lots in the next few months to better understand this. But so this is a disclaimer that this is what we know right now. Okay, so I'll start out with some easy ones, okay? COVID-19 is caused by, is it a bacteria, a virus, a pathogen that was created in a lab in China, a pathogen that came from a bat, a pathogen that's related to the common cold virus, or a flu virus? More than one answer can be right. Okay, so the answers are, it's not a bacteria, it is coronavirus, so that was an easy one. It was not created in a lab in China, but it appears to have been come from a bat population that the virus was in, that then had some genetic changes in it that allowed it to enter into the human population. It is a pathogen related to the common cold virus, and I really think this is going to be very interesting and very important information coming out from this, because it's in the same family as 4 other coronaviruses that just are circulating all the time in all of our communities around the world. And we're wondering if the reason why we're not seeing as many infections or serious infections in young kids is perhaps that those common cold viruses may be providing some protection from COVID. And it is not a flu virus, even though it may feel like the flu and seem like the flu or act like the flu, it's actually not a flu virus. And the COVID-19 is actually caused by SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. Okay, so what are the most likely ways you can get infected with SARS-CoV-2? Packages from China, a wedding, your pets, a concert, contaminated shoes, a swimming pool, a bar, your friends. Well, the most common ways are really with large groupings or even close groupings with small numbers of people, okay? So you're unlikely to get it from a package from China. It will not stay on packages very long, and that's just really not a very efficient way to transmit it. Same with contaminated shoes. You also won't get it from a swimming pool, but if you're at a crowded swimming pool and you have increased contact with other people, you certainly can get it person to person, okay? Your pets, they're doing lots of studies now to look at pets. Pets can get it in their nose, but it doesn't really live in them very, very easily or very well, and they're not, have not been shown to be important transmitters of the infection. Concerts, bars, hanging out with your friends, that's how it gets transmitted. Okay, COVID-19 transmission, which statements are true? I can't transmit COVID-19 to someone if I don't have cold symptoms. I don't have a fever. I wasn't face-to-face with someone for at least 10 minutes. If I'm wearing a face mask, if I'm wearing rubber gloves. Which statements are true? Actually, none of them are true. You don't have to have cold symptoms or fever. Asymptomatic transmission is very common and is felt to be a very effective way of transmitting COVID. You can spend a very short period of time with someone, especially if you have no face mask, and spread COVID-19. You are wearing a face mask. Face masks can decrease the risk, but are not 100% effective, okay? Definitely decreases the risk. It's definitely worth doing, but it's not 100%, okay? And you can transmit COVID-19 to someone if you were wearing rubber gloves. I always love when I go through my Chick-fil-A and they have the gloves and they're, you know, protecting themselves, and then they take my credit card, and then after that, they put their hands, use my credit card and itch their nose or itch their eyes. It's really not doing them any good, okay? It's not doing me any good, okay? You still have to have good hand washing and be careful with where you are putting your hands. The very effective ways of transmitting COVID is by touching your eyes, nose, or mouth with your contaminated fingers that have been exposed to COVID. Okay, COVID-19 disease severity. Which statements are true? COVID-19 is the deadliest virus in the world. COVID-19 is more severe than influenza. Only old people or people with medical problems get severe disease. Young children don't get sick with COVID-19. Most people get better with COVID-19. Okay, so I show families here because, you know, everybody in that family is at risk for getting COVID if they're exposed to it. I want everybody to remember that, okay? And there are some people in that family that are going to be riskier than others, okay, for either getting the infection or having more severe disease. So here's the answers to that. COVID-19 is not the deadliest virus in the world. It may feel like it right now, but it really is not. There are many other viruses that are much more deadly than coronavirus. COVID-19 is more severe than influenza, for sure. People of any age can have severe disease, but older people or people with medical problems get severe disease more often. Young children can get very sick with COVID-19. I've taken care of many of those kids in the hospital already this year who have been very, very sick with COVID. But importantly, most people will get better after they've been infected with COVID-19. Okay, which of these are effective treatments for COVID-19? Or I should say, have some effectiveness to COVID-19. And this is probably the slide that could change the most over the next few months, because we probably will have more information on some of these and many other products that will come down the road. Okay, gargling with mouthwash. Okay, is that going to treat you? If you know that you've just been exposed to somebody and you go home and you gargle with mouthwash, are you going to, is that going to treat that COVID that you just got exposed to? The anti-malarial hydroxychloroquine, the antibiotic azithromycin, sucking on a lemon. Will that acid in that lemon get rid of that COVID that you've just been exposed to? Drinking alcohol, pouring alcohol on you, doing anything with alcohol. Will that be an effective treatment for COVID-19? The antiviral remdesivir, is that effective? UV light, in convalescent plasma, where they basically take plasma from somebody that's had, take blood from somebody that has had COVID infection, see if they have antibody to it, and they take their plasma and give that to other people to help boost their antibody. So the only one of these that has been shown to have some effectiveness, and it's even not that terrific, is the antiviral remdesivir. There are lots of studies on the way, and there may be other things down the road that may be effective, but at this point, this is all we have. I should mention, and I should have put on the slide, is steroids have also shown to be effective in treating COVID as well. These are not cures, and if you give both the remdesivir and the steroids, that doesn't guarantee that you are going to recover from it. But the people that get these two products, these two drugs, are more likely to have a little better outcome than if you don't get them. Okay, this is important. Okay, how can you tell if you're infected? Okay, you can do a cough test. If you hold your breath for 10 seconds and you don't cough, then you don't have COVID. A thermal scanner. If you get a thermal scan and you don't light up, you don't have COVID. A saliva PCR for COVID-19. If you get your saliva tested and the test is negative, you don't have COVID. Well, all of these are false. Holding your breath doesn't really tell you much of anything other than that you can hold your breath, okay? Some people just don't hold their breath well. It really is not indicative of anything. I think you could even have asthma and not be able to hold your breath. It's very non-specific if for whatever reason you can't hold your breath. It doesn't have anything to do with COVID. A thermal scanner only tells you if you have a temperature elevation and not COVID. And the saliva PCR, this only detects COVID-19 in very, very symptomatic people and won't detect COVID-19 early in the infection or if there are minimal symptoms. Okay, so it might be good if you're really sick already, okay, it might tell you if you have it, but it misses it at the beginning and the end of the infection or if you don't have very many symptoms. So the respiratory PCR is how we diagnose COVID-19. You can do a nasal swab that's done really well, done one swab on both sides, or a nasopharyngeal swab, which is one that is done really far back in the back of your nose because very high up is uncomfortable. And you want to make sure that the test is done with an approved COVID-19 PCR test. The tests are not 100% accurate. They're really very, very good, okay, but they're not 100%. And people can be intermittently positive. So if I got exposed to somebody today and I said, oh my goodness, I better go get tested, I'm not likely to be positive for a few days, okay? So I would, what typically happens is we recommend people wait for a few days and then get tested, because if you have it, it takes time for the virus to incubate and to replicate in your nose and to become positive. And so if I got it infected today, I'm not going out today to get a test. I will wait till Monday or Tuesday, 3 days, 4 days to get tested, okay? And even people that have it, we've had people that have been in the hospital that one test is positive one day, it's negative the other day. We don't know why, and then it's positive again the next day, okay? So we don't know why that happens, but there seems to be some intermittent shedding of this virus that isn't really what we typically see with other viruses. The respiratory PCR is the best test. There are other tests called antigen tests, not actually looking for the virus or just looking for a virus antigen. They are not as good. They can have what we call false positives. They can give you a positive test when you don't really have it, and false negatives. They can miss the infection when you have it. Okay, so you definitely would go for that respiratory PCR when you can. COVID-19 antibody test. Which statements are true? If my antibody test is positive, I can't get infected again. I can't transmit COVID-19 to others. I don't have to wear a mask anymore, and I can do whatever I want to do. Well, all statements are false, okay? And that might be surprising to some of you, because I think a lot of times people are like, once I have the antibody, I'm good to go. I am not going to get infected again. I'm going to be fine. This is, this is my, get out of jail free card here. I can do whatever I want. And that's really not true. We don't really have a very good understanding of what a positive antibody test means at this point. We may in a few months, but right now we don't. At this point, our understanding is if your antibody test is positive, you can still get infected again. You can still transmit COVID-19 to others. You still need to wear a mask, and you really can't do whatever you want to do. At this time, we're studying to understand what a positive antibody test means. Does it mean you're protected at all? We're really not sure. If it does provide protection, we don't think it's going to be very long lasting. Even if your antibody test is positive, you can still transmit COVID-19 because the antibody can become positive very quickly while you're just infected. Okay, so we have people that we do an antibody test on at the same time they're doing the PCR, and both can be positive. So that does not tell you anything. Okay, so we're doing lots and lots of studies to better understand, you know, how we can use this test. But right now, honestly, it's not very useful from a clinical standpoint and how to provide guidance for people that have a positive test. Okay, what are good ways to decrease your risk for COVID-19? Eating garlic, social distancing, hand washing, using hand sanitizer, using masks, taking vitamin C. So I think everybody would have gotten this right. Eating garlic, it has some good things about it from a health standpoint, but is not going to decrease your risk for COVID. Neither is vitamin C. But social distancing, hand washing, using hand sanitizer, and using masks have all been shown to decrease your risk for infection. Last, I want to talk a little bit about vaccines to prevent COVID-19. Which statements are true? COVID-19 vaccine will be more effective than getting a natural COVID-19 infection. One COVID-19 vaccine shot will protect me for life. A flu vaccine will protect me from COVID-19. One risk for COVID-19 vaccines is that you can get COVID-19 from that vaccine. And then last, COVID-19 vaccines will be widely available for everyone before the end of the year. So none are true. Okay, a COVID vaccine will not be more effective than a natural COVID-19 infection. Natural infection provides more protection than vaccines. One COVID-19 vaccine shot will not protect you for life. With endemic coronaviruses, those 4 coronaviruses that we just have in our life, and we have had them forever, that we get every year, you can get repeat infections throughout your life, even in the same year. Therefore, one dose of vaccine is unlikely to provide any kind of long-term infection. A flu vaccine will not protect you from a COVID-19 infection. It's going to provide you with some protection for flu, which is important because then that takes out one more infection that you can get during the respiratory season, but it's not going to protect you from COVID. And you will not get COVID-19 from the vaccine. None of the vaccines are live viral vaccines at this point, so this is not possible. And COVID-19 vaccines will not be widely available for everyone by the end of 2020. If vaccines are available in 2020, it will only be for small numbers of people in selected groups. So hang in there. We're going to get through this. Wear your mask to protect yourselves and others. Do your best to social distance. Limit big gatherings. If you're infected, quarantine yourself. Do what is asked of you when the health department calls, when the school asks you to quarantine yourself. Do your best with that. Okay, you're gonna prevent infection of other people if you can quarantine yourself. Follow the science to get the answers. Okay, really listen to what the scientists are saying. Everybody's doing their best to figure this out. It's going to get better, but it's going to take a little while longer before we get there.
We do have a couple questions, Dr. Staat.
Yep.
The first is, many masks don't fit well. What about when you have to adjust your mask? Do you need to wash your hands or change the mask? Is there a relationship between vitamin D and the severity of COVID? Those are the 2 questions.
That's great. So I think I'll start with the vitamin D question. I think there are lots of factors that are somehow making a difference in who gets sick and who doesn't get sick, okay? And who gets the infection, who doesn't get the infection. And I think some of these factors may be vitamin D. I don't think it's the only thing, okay, but it's probably, it could have some role. It probably also your genetics, your underlying diseases. There are lots of different factors that I think could play a role. There were lots of early data out about blood group. You know, if you were type A or type O, you were less likely to have severe disease, okay? That's really early data. There's still lots and lots of work to be done to understand about these risk factors, and everybody's working very hard to understand it. And vitamin D, I would say, would be in that same category. As far as masks, you definitely want the best fitting mask that you can have, okay? If you are really feeling air going outside of your mask, like fogging up your glasses and you're blowing air up into your eyes, then that's air that's getting out into the, past your mask, that's not going through the mask, that could infect others. If you need to adjust your mask, because you don't know oftentimes if you're infected or not, and you're trying to really control your secretions, then you absolutely should wash your hands after you have adjusted the mask. Anytime you're having your hands in your nose and your mouth and your eyes, you should be washing your hands for 20 seconds. Okay, just sing the ABC song while you're washing your hands, or using the hand sanitizer. So that's a great question, and I think unless you videotape yourself or see people videotape and see how often we have our fingers on our face, you don't really realize how much we expose ourselves all the time with those behaviors.
All right. Well, thank you for an amazing and informative presentation. I hope you have a wonderful weekend.
Thank you.
Take care. Bye.
Bye-bye.